Trees feel familiar because they are everywhere. They line streets, fill parks, and form forests that cover approximately 30% of Earth's land surface. From a human perspective, trees appear as individuals, rooted in place, growing slowly, existing separately from one another. Yet, when examined more closely, that sense of separation begins to soften. Trees communicate. They share resources. They recognize relatives and adjust their behavior accordingly. Beneath the forest floor exists a network of connections so complex that scientists have dubbed it the Wood Wide Web, a system linking individual trees into communities that function in ways that challenge simple definitions of individuality. Welcome to Sleep Knowledge. If you find comfort in exploring the quiet complexity of the natural world, settle in and let these 100 facts about trees unfold slowly. And if you feel moved to, share where you are drifting off tonight. It is always warming to know how far these gentle thoughts travel. These are not metaphors or speculation. The phenomena described here emerge from decades of careful research in forest ecology, plant physiology, and mycology. Trees possess capabilities documented through controlled experiments, field observations, and technologies that allow scientists to trace the movement of nutrients and signals through forest ecosystems. Understanding trees requires patience. Their lives unfold across centuries rather than years. Their responses to environmental changes occur slowly, sometimes imperceptibly. A tree injured today might adjust its growth pattern over decades. A seed germinating this year might not reach reproductive maturity for 50 years or more. There is something calming in that slowness. Trees demonstrate that not everything in nature operates at human timescales. They show that growth can be gradual, that connections can form slowly, and that patience represents not passivity, but a strategy refined across millions of years of evolution. Settle in and let the world of trees unfold gently. Beneath every forest floor exists a network more complex than it appears. Fungal threads called hyphae, finer than human hair and measuring in micrometers rather than millimeters, grow through soil in all directions. These threads connect with tree roots, forming structures called mycorrhizae, literally fungus root in Greek. This relationship was identified more than a century ago, but its full implications remained unclear until recent decades. In 1997, forest ecologist Suzanne Simard at the University of British Columbia demonstrated that these fungal networks could transfer carbon between trees. She injected carbon isotopes into paper birch trees and detected those isotopes appearing in nearby Douglas firs, connected through mycorrhizal networks. The mechanism is straightforward. Trees produce sugars through photosynthesis. Some of those sugars move to roots, where mycorrhizal fungi access them. The fungi use those sugars to grow their networks through the soil, and in return, they transport nutrients they collect, phosphorus and nitrogen particularly, back to tree roots. Both organisms benefit. What makes the system remarkable is its scale. A single fungal individual can connect with multiple trees, potentially dozens or hundreds, depending on the species and environment. These networks create pathways through which not just nutrients, but chemical signals can travel from tree to tree. Research has shown that when one tree experiences insect attack, neighboring trees connected through mycorrhizal networks sometimes increase their production of defensive compounds, even before they themselves are attacked. The mechanism remains under investigation, but the pattern has been documented repeatedly. Information appears to move through forests faster than would be expected if trees communicated only through airborne chemicals. Some studies suggest trees may recognize their relatives through these networks, allocating more resources to offspring than to unrelated neighbors. Carbon isotope tracing experiments found that Douglas firs transferred more carbon to closely related Douglas fir seedlings than to more distantly related trees of the same species. The mechanism of this recognition remains unknown, but the behavior pattern has been observed in multiple forest systems. The fungal partners in these networks are not passive conduits. They retain approximately 30% of the sugars they transport from trees, using that energy to maintain and expand their networks. In return, they provide access to nutrients that tree roots alone cannot reach efficiently. The relationship represents mutual benefit, though the balance can shift depending on environmental conditions. Older trees, sometimes called hub trees or mother trees, typically possess more mycorrhizal connections than younger trees. Their established root systems extend deeper into soil, accessing water and mineral resources that seedlings cannot yet reach. Through mycorrhizal networks, these mature trees can redistribute resources, effectively supporting younger trees during their establishment phase. This changes how forests are understood. Rather than collections of competing individuals, forests function partly as cooperative networks where established trees provide support to developing ones. The cooperation is not altruistic in a conscious sense, but represents evolutionary strategies that improve overall forest survival, which in turn improves individual tree survival. Not all fungi form mycorrhizal associations. Different tree species associate preferentially with different fungal groups. Oak and pine roots typically host ectomycorrhizal fungi that form sheaths around root tips. Maple and cedar prefer arbuscular mycorrhizal fungi that penetrate root cells directly. These different associations create different network structures, with ectomycorrhizal networks typically forming more extensive soil webs than arbuscular networks. The discovery of these networks required sophisticated techniques. Fungal hyphae are delicate and nearly invisible in soil. Tracking nutrient movement requires isotopic labeling. Confirming that two trees share a fungal connection requires genetic analysis to determine whether fungal samples from different locations belong to the same individual organism. What has emerged from this research is recognition that forests operate through layered connections. Above ground, trees compete for sunlight. Below ground, they participate in resource exchange networks facilitated by fungi. Both competition and cooperation operate simultaneously, creating complex ecosystem dynamics that simple models of plant behavior cannot capture. Understanding mycorrhizal networks does not romanticize forests or attribute human-like emotions to trees. It reveals that plant behavior emerges from biochemical signaling systems refined through evolution, systems capable of responding to environmental conditions in ways that appear sophisticated when observed carefully. Trees are not conscious in human terms, but they are responsive, adaptive, and interconnected in ways that continue surprising researchers. Methuselah, a bristlecone pine growing in California's White Mountains, germinated approximately 4,857 years ago. This tree was already centuries old when the first Egyptian pyramids were built. It continued growing through the rise and fall of countless human civilizations. It remains alive today, adding annual rings to its trunk each growing season, just as it has for nearly 5 millennia. Bristlecone pines live longer than any other non-clonal tree species. The oldest known living bristlecone, found in the same forest as Methuselah, but kept secret to protect it from vandalism, approaches 5,100 years of age. These trees have witnessed climate changes spanning the entire period of recorded human history, and substantial portions of the period before humans developed writing. Bristlecone pines achieve such longevity through characteristics that seem counterintuitive. They grow extremely slowly, in harsh, high-elevation environments where most other trees cannot survive. Cold temperatures, short growing seasons, poor soils, and high winds create conditions that stress most organisms. Yet these stresses contribute to bristlecone longevity. Slow growth produces dense wood that resists decay. The wood contains high concentrations of resins that protect against insects and fungi. In extreme conditions, bristlecones sometimes die back partially, maintaining only a narrow strip of living tissue connecting roots to a few branches. This sectored architecture allows the tree to survive, even when most of its structure is dead. Tree longevity varies dramatically across species. Some tropical trees live less than a century. Temperate forest trees like oaks and maples commonly reach 200 to 400 years. Conifers in harsh environments, including bristlecones, can persist thousands of years. The oldest confirmed tree ever documented was a bristlecone called Prometheus, cut down in 1964, which was determined to be at least 4,862 years old. Clonal trees can persist even longer than individual trees. Pando, a grove of quaking aspens in Utah, consists of approximately 47,000 stems connected by a single root system. This clonal organism is estimated to be 80,000 years old, though individual stems live only 100 to 150 years. The root system persists, continuously producing new stems as old ones die. Age determination in trees relies primarily on dendrochronology, the science of counting and analyzing annual growth rings. Each year, trees in seasonal climates produce a distinct ring, consisting of light-colored spring growth and darker summer growth. Counting these rings provides an accurate age estimate for trees. However, ring counting has limitations. Some tropical trees do not form distinct annual rings because they experience no winter dormancy. Some trees form multiple rings in a single year under stress conditions. Missing rings can occur during extreme drought years when no growth occurs. Cross-referencing ring patterns from multiple trees helps dendrochronologists account for these complications. Tree rings contain information beyond age. Ring width indicates growing conditions each year, with wider rings forming during favorable years and narrow rings during stressful years. Chemical analysis of rings reveals past atmospheric conditions, including pollution levels and volcanic eruptions. Stable isotope ratios in wood reflect temperature and precipitation patterns during growth years. This makes old trees valuable archives of environmental history. Bristlecone pine rings extend records of temperature and precipitation back thousands of years in regions where human records exist only for the most recent centuries. These long records help climate scientists understand natural variability and distinguish it from human-caused changes. The oldest living trees witnessed dramatic climate shifts. Methuselah germinated during the warm period following the last ice age. It has experienced multiple cold periods and warm periods since then, each lasting centuries. Its rings record droughts, floods, volcanic eruptions, and countless seasonal cycles. Yet the tree persists, demonstrating remarkable resilience. Studying ancient trees requires care. Taking core samples with increment borers allows age determination without killing trees, though repeated sampling can stress them. Conservation of old-growth forests protects not just individual ancient trees, but also the ecosystems they help maintain. Once ancient trees are removed, centuries or millennia pass before similar individuals can develop. Trees produce oxygen through photosynthesis, but the amount varies dramatically depending on tree size, species, and growing conditions. A single large tree might produce enough oxygen annually to support 2 people. Tropical rainforest trees in ideal conditions produce more oxygen per unit biomass than temperate or boreal species, experiencing seasonal limitations. However, focusing only on oxygen production misses trees' broader ecological roles. Forests influence water cycles, stabilize soils, moderate local temperatures, provide habitat for countless species, and store carbon. These functions often matter more than oxygen production for ecosystem health and human well-being. The oxygen trees produce comes from splitting water molecules during photosynthesis. Light energy powers the reaction that separates water into hydrogen and oxygen. The hydrogen combines with carbon dioxide to form sugars. The oxygen releases into the atmosphere as a byproduct. This process repeated in trillions of leaves across billions of trees, maintains atmospheric oxygen at approximately 21%. Before photosynthetic organisms evolved, Earth's atmosphere contained almost no free oxygen. Early photosynthetic bacteria began releasing oxygen as a waste product approximately 2.4 billion years ago. This fundamentally changed Earth's chemistry and atmosphere, allowing oxygen-breathing organisms to evolve. Today, most atmospheric oxygen production occurs in ocean phytoplankton rather than terrestrial plants, though both contribute significantly. Trees also consume oxygen through respiration. Like all living organisms, trees break down sugars to release energy, consuming oxygen and producing carbon dioxide. This respiration occurs continuously, though photosynthesis only occurs during daylight hours, when sufficient light is available. The net oxygen balance for a healthy growing tree is positive, meaning more oxygen is produced than consumed. Mature forests approach oxygen balance, where production roughly equals consumption. Young growing forests produce net oxygen as they accumulate biomass. Old-growth forests with stable biomass may produce little net oxygen, though they provide other crucial ecosystem services. When trees die and decompose, their stored carbon oxidizes, consuming oxygen. The relationship between trees and atmospheric composition extends beyond oxygen. Trees remove carbon dioxide from the atmosphere during photosynthesis, converting it into plant tissue. This carbon storage, called carbon sequestration, helps moderate atmospheric carbon dioxide concentrations and therefore climate. A single tree stores carbon equivalent to several tons of carbon dioxide over its lifetime, depending on size and species. Large tropical trees can store more than 100 tons of carbon dioxide equivalent. Even small temperate trees store substantial carbon in wood, branches, and roots. When trees die naturally and their wood decomposes slowly, much of this carbon returns gradually to soils rather than immediately to the atmosphere. Forests globally store approximately 861 gigatons of carbon, representing about 1/3 of all carbon stored in terrestrial ecosystems. This carbon exists in living biomass, dead wood, leaf litter, and soil organic matter. Protecting existing forests prevents this stored carbon from releasing rapidly into the atmosphere. Establishing new forests on degraded lands can sequester additional carbon. However, carbon storage in forests is not permanent. Fire, disease, insect outbreaks, and harvest all release stored carbon. Climate change itself threatens forest carbon storage by increasing stress, mortality, and disturbance frequencies. Managing forests for carbon storage requires balancing growth that accumulates carbon against risks that could release it. Trees influence climate through mechanisms beyond carbon and oxygen. Forests affect albedo, the reflectivity of Earth's surface. Dark forest canopies absorb more solar radiation than lighter surfaces like grasslands or ice, warming local and regional climates. This albedo effect is most pronounced in boreal regions, where forests replace snow-covered ground. Forests also affect water cycles through transpiration. Trees move water from soil through roots, stems, and leaves into the atmosphere. A single large tree can transpire hundreds of gallons of water daily during peak growing season. This water vapor contributes to cloud formation and precipitation, creating feedback loops where forests help generate their own rainfall. The Amazon rainforest demonstrates this water cycling dramatically. Studies suggest that forests produce more than half of regional rainfall through transpiration and water recycling. Deforestation reduces this moisture recycling, potentially shifting the region toward drier conditions that cannot support forest, creating a concerning positive feedback. Trees communicate through multiple channels beyond mycorrhizal networks. When insects attack leaves, trees emit volatile organic compounds into the air. Neighboring trees detect these airborne chemicals and respond by increasing production of defensive compounds, preparing for potential attack even before insects arrive. This aerial communication operates independently of root contact or fungal networks. The specific chemicals released vary depending on the attacking insect species, allowing receiving trees to tailor their defensive response to the specific threat. Some trees even release compounds that attract predatory insects, effectively calling for help against herbivores. The timing matters. Trees that detect warning signals and prepare defenses before attack suffer less damage than trees attacked without warning. This provides evolutionary advantage to both signaling and receiving individuals in populations where neighbors are genetically related. However, the benefits decrease as distance increases because chemical signals dilute with distance. Physical connections also facilitate communication. When tree branches touch, they sometimes form natural grafts where cambium layers fuse. These grafted connections can transport water and nutrients between trees. Root grafts form similarly underground, where roots of the same or related species grow in contact. These physical connections create additional pathways for resource sharing beyond mycorrhizal networks. Trees sense their environment through multiple pathways. Light receptors called phytochromes detect light quality and quantity, allowing trees to adjust growth patterns based on light conditions. Red light indicates open sky above, while far red light indicates shade from other plants. This information guides branch and leaf placement to maximize light capture. Gravity sensing occurs through specialized cells containing starch grains that settle to the bottom of cells, indicating direction. When trees tip, this gravity sensing triggers corrective growth that straightens the trunk. The response can take years to complete. But old trees growing on steep slopes demonstrate remarkable ability to maintain vertical growth despite underlying ground angle. Touch sensing allows trees to respond to physical stimuli. Trees in windy locations develop shorter, thicker trunks than sheltered trees of the same species. This thigmomorphogenesis represents an adaptive response to mechanical stress, producing structures better able to withstand wind loading. Even gentle, regular touching of seedlings can trigger this response. Chemical sensing allows trees to detect soil nutrient concentrations and adjust root growth accordingly. Roots grow preferentially toward higher nutrient concentrations. When nutrients become depleted locally, trees increase root growth in other directions, or increase mycorrhizal fungal associations to access nutrients from larger soil volumes. Temperature sensing triggers seasonal responses. Deciduous trees track cumulative cold exposure during winter, using this information to time spring bud break appropriately. Too early and spring frost can damage tender new growth. Too late and the tree misses valuable growing days. Timing must adapt to local climate patterns, which is accomplished through genetic adaptation and phenotypic plasticity. Water stress sensing occurs through hormone signaling. When roots experience drought, they produce abscisic acid, a hormone that travels through the tree to leaves. Leaves respond by closing stomata, reducing water loss through transpiration. This reduces photosynthesis, but prevents catastrophic water loss that could kill the tree. These sensing systems demonstrate that trees, while lacking nervous systems or brains, possess sophisticated mechanisms for perceiving and responding to their environment. The responses are slower than animal responses, operating on timescales of hours to days rather than milliseconds to seconds. But the capability to sense, process information, and respond adaptively is clearly present. Tree growth follows predictable patterns but shows remarkable variation depending on species and environment. Apical meristems at branch tips produce new length growth. Lateral meristems called cambium produce new diameter growth. These meristems contain undifferentiated cells capable of dividing to produce new tissues. Height growth typically follows a sigmoid pattern. Young trees grow slowly while establishing root systems. Growth accelerates during middle age, when trees have sufficient roots to support rapid height increase, but have not yet reached size, where mechanical limitations restrict growth. Growth slows again as trees approach maximum height for their species and environment. Maximum height varies dramatically. Some tropical species exceed 100 meters. Coastal redwoods are the tallest trees, with the record holder, Hyperion, measuring 115.92 meters. Temperate hardwoods like oaks typically reach 30 to 40 meters. Height is limited by water transport physics. Lifting water from roots to leaves requires overcoming gravity and friction. Eventually, the energy required exceeds what photosynthesis can supply. Diameter growth continues throughout a tree's life, though the rate slows with age. Trees add one ring annually in seasonal climates. Ring width depends on growing conditions, with favorable years producing wide rings and stressful years producing narrow rings. Extremely old trees may add rings only micrometers thick during harsh years. Root systems are less visible but equally important. Roots anchor trees, preventing toppling. They absorb water and nutrients from soil. They store energy reserves that support leafing out each spring in deciduous species. Root system size often exceeds canopy size, spreading laterally well beyond branch spread. Root depth varies by species and soil conditions. Most tree roots occupy the top meter of soil, where nutrients and oxygen are most abundant. However, some species send taproots much deeper to access groundwater during drought. Desert trees can root more than 50 meters deep, reaching stable water sources below seasonal variation. The transition from seed to mature tree spans decades to centuries. Seeds germinate when conditions are favorable, typically requiring adequate moisture, appropriate temperature, and sometimes specific light conditions. The germinating seed uses stored energy reserves to produce initial roots and shoots before photosynthesis begins. Seedling establishment is the most vulnerable life stage. Competition from existing vegetation, herbivore browsing, drought stress, disease, and shading all cause high seedling mortality. In many forests, less than 1% of seeds survive to become established trees. This high mortality is why mature trees produce such enormous seed quantities. Saplings begin rapid growth once established, competing for light and resources. Canopy gaps created by falling trees provide opportunities for saplings to accelerate growth and reach the canopy. In closed forests, saplings may persist for decades in shade, growing slowly, waiting for disturbance to create light opportunities. Young trees allocate resources differently than mature trees. Young trees invest heavily in height growth to reach light. Mature trees invest more in reproduction, producing seeds and supporting extensive root and branch systems. Old trees may allocate more resources to defense and maintenance as growth slows. Tree reproduction occurs sexually through seeds and sometimes vegetatively through cloning. Sexual reproduction provides genetic variation, allowing adaptation to changing conditions. Seeds can disperse long distances, colonizing new areas. However, seed production is costly, requiring substantial energy and nutrients. Vegetative reproduction through root sprouting or branch layering allows rapid colonization of disturbed areas. Aspen clones spread through root suckering, producing genetically identical stems across large areas. Willow branches that fall in water can root and establish new individuals. These cloning strategies work well in stable environments, but provide less genetic variation than sexual reproduction. Flowering trees produce flowers to attract pollinators or facilitate wind pollination. Flowers require significant resource investment. Some trees flower annually. Others, particularly temperate species, show mast seeding years where synchronous heavy seed production occurs across regions. These mast years overwhelm seed predators, ensuring some seeds survive to germinate. Conifers produce cones instead of flowers. Pollen cones release enormous quantities of pollen grains that wind carries to seed cones. Seed cones protect developing seeds until they mature. Cone opening and seed release are often triggered by environmental cues like fire or prolonged drying, timing dispersal for favorable conditions. Seed dispersal strategies vary dramatically. Wind dispersal works for lightweight seeds with wings or plumes, allowing travel hundreds of meters from parent trees. Gravity dispersal works for heavy seeds, though they typically remain near parent trees. Animal dispersal occurs when fruits attract animals that consume seeds and deposit them elsewhere in droppings. Each dispersal strategy has advantages and disadvantages. Wind dispersal covers large areas but provides little control over where seeds land. Most land in unsuitable locations. Animal dispersal is more targeted, as animals often deposit seeds in favorable locations, but it requires substantial investment in fruit production. Gravity dispersal requires minimal investment but limits range expansion. Fire plays complex roles in forest ecology. Many tree species have evolved fire adaptations. Thick bark protects cambium from brief ground fires. Serotinous cones open only after fire, releasing seeds into nutrient-rich ash beds cleared of competition. Root systems that survive fire can resprout, rapidly recolonizing burned areas. However, fire frequency and intensity matter. Frequent low-intensity fires maintain open forests adapted to fire, preventing fuel accumulation that could cause catastrophic burns. Fire suppression allows fuel accumulation, potentially leading to intense fires that kill even fire-adapted trees. Understanding natural fire regimes helps inform management of fire-prone forests. Climate change is altering fire patterns in many regions. Longer fire seasons, drier conditions, and more extreme weather increase fire frequency and intensity. Trees adapted to historical fire patterns may face conditions exceeding their tolerance. This represents one of many ways climate change challenges forest persistence. Insects interact with trees in countless ways. Some insects pollinate flowers, providing essential reproductive services. Others consume leaves, reducing photosynthesis and weakening trees. Bark beetles bore through bark and cambium, disrupting water and nutrient transport. Wood-boring beetles consume dead wood, accelerating decomposition. Trees have evolved multiple defense mechanisms against insect herbivores. Physical defenses include tough leaves, waxy coatings, thorns, and spines. Chemical defenses include toxic or distasteful compounds that discourage feeding. Induced defenses activate after attack begins, ramping up chemical production in response to damage. However, insects evolve counter-adaptations. Some species detoxify defensive chemicals. Others specialize on trees with specific defenses, turning defensive compounds to their advantage. This ongoing evolutionary arms race between trees and insects has produced remarkable diversity in both groups. Bark beetles in particular can cause massive tree mortality during outbreak periods. Warming temperatures allow beetles to complete more generations per year and expand into previously unsuitable habitats. Drought stress weakens trees' ability to produce resin that normally drowns attacking beetles. These factors combine to cause unprecedented bark beetle outbreaks across western North America. Pathogenic fungi and bacteria also affect trees. Root rot fungi kill fine roots, impairing water and nutrient uptake. Canker diseases create dead patches on stems and branches. Wilt diseases clog water-conducting vessels, causing branches or entire trees to die. Dutch elm disease and chestnut blight have killed billions of trees across North America. Trees defend against pathogens through physical barriers like bark and chemical compounds that inhibit pathogen growth. Compartmentalization walls off infected tissue, preventing pathogen spread. However, these defenses are not always effective, particularly against novel pathogens, to which trees have no evolved resistance. Introduced pests and pathogens pose particular threats. Emerald ash borer, an Asian beetle introduced to North America, has killed hundreds of millions of ash trees. The trees have minimal defenses because they evolved without this pest. Such biological invasions represent growing threats to forests in an era of global transportation. Birds, mammals, and other animals depend on trees for food and habitat. Fruits and nuts provide high-energy food. Cavities in trunks and branches offer nesting and roosting sites. Bark crevices harbor insects that insect-eating birds consume. Canopy structure creates complex three-dimensional habitat unavailable in grasslands or shrublands. Trees benefit from some of these relationships. Fruit-eating animals disperse seeds. Insect-eating birds reduce herbivore populations. Predators that hunt from tree perches may help reduce populations of small mammals that consume tree seeds and seedlings. These mutualisms contribute to forest ecosystem function. Symbiotic relationships extend beyond mycorrhizal fungi. Nitrogen-fixing bacteria in root nodules provide accessible nitrogen to some tree species. These bacteria convert atmospheric nitrogen into ammonia that trees can use. In return, trees provide carbohydrates to bacteria. This symbiosis allows trees to thrive in nitrogen-poor soils where other species struggle. Lichens, composite organisms of fungi and algae or cyanobacteria, grow on tree bark. These are not parasitic. The tree provides a substrate, while lichens neither harm nor directly benefit the tree. However, lichens contribute to forest ecosystems by fixing nitrogen and providing food for various animals. Their presence indicates clean air. Tree diversity within forests creates ecological complexity. Different tree species occupy different niches, exploiting resources in different ways. Shade-tolerant species thrive in forest understories. Light-demanding species colonize gaps. Different rooting depths, phenologies, and resource requirements allow multiple species to coexist. This diversity provides resilience. Diverse forests often resist pest and disease outbreaks better than monocultures because specialized pests cannot spread easily when host trees are scattered among non-host species. Diversity also provides functional redundancy, so if one species declines, others may compensate. However, not all forests are diverse. Boreal forests are often dominated by just a few conifer species. Temperate deciduous forests typically contain a dozen or two tree species. Tropical rainforests may contain hundreds of tree species per hectare. This latitudinal diversity gradient represents one of ecology's most consistent global patterns. Multiple factors contribute to tropical diversity. Stable tropical climates allow specialization. High productivity supports larger populations. Large tropical regions provide more evolutionary opportunities. Absence of ice age disruptions allowed long-term species accumulation. The relative importance of these factors remains debated. Conservation of tree diversity faces challenges. Many tropical tree species are rare, known from only a few individuals or small populations. Habitat loss threatens these species with extinction before they are even scientifically described. Documenting and protecting tropical tree diversity remains an urgent priority. Trees modify local and regional climates. Urban trees reduce temperatures through shade and evapotranspiration. A single large tree can cool its immediate surroundings by several degrees on hot days. Street trees make neighborhoods more comfortable for residents while reducing building cooling costs. Forests, moderate temperature extremes compared to open land. Forest interiors experience less daily temperature variation than adjacent clearings. This buffering creates relatively stable microclimates that many species require. Clear cutting eliminates this buffering, exposing soils and remaining vegetation to more extreme conditions. The scale of these climate modifications can be substantial. The Amazon rainforest influences precipitation patterns across South America. Deforestation in the Amazon could reduce rainfall across the continent, affecting agriculture in regions far from the forest. Boreal forests influence albedo at continental scales, affecting global climate. Trees also reduce wind speeds. Forest edges and windbreaks slow wind, reducing soil erosion and creating more favorable conditions for crops and animals. Traditional agricultural landscapes often include trees for this windbreak function. Though, Modern industrial agriculture has removed many such trees. Soil relationships are fundamental to tree ecology. Tree roots stabilize soil, preventing erosion on slopes and stream banks. Root systems create pathways for water infiltration, reducing surface runoff. When trees are removed, soils become vulnerable to erosion, particularly on steep terrain or during heavy rainfall events. Trees also build soil through leaf litter and root turnover. Deciduous trees drop leaves annually, adding organic matter to soil. Fine root turnover occurs continuously, as small roots die and new ones grow. This organic matter input feeds soil decomposer communities and increases soil carbon storage. Different tree species affect soil chemistry differently. Conifers typically produce acidic leaf litter that lowers soil pH. Broad-leaved trees often produce less acidic litter. Nitrogen-fixing trees enrich soil nitrogen. These differences mean that tree species composition influences soil properties, which in turn influences what plant species can grow. Tree roots interact intensively with soil organisms. Beneficial fungi and bacteria help trees access nutrients. Pathogenic organisms attack roots. Herbivorous insects consume root tissue. Predatory organisms hunt these herbivores. The soil food web connected to tree roots contains remarkable diversity and complexity. Mycorrhizal fungi are not the only important root symbionts. Endophytic fungi live within root tissues without causing disease. Some produce compounds that protect against pathogens. Actinomycetes bacteria in soil produce antibiotics that suppress pathogenic organisms. These beneficial associations help explain why trees grown in sterile soil perform poorly compared to trees in natural soil containing diverse microbial communities. Nutrient cycling in forests operates through multiple pathways. Trees absorb nutrients from soil. These nutrients become incorporated into leaves, wood, and roots. When leaves fall and organisms die, decomposers break down organic matter, releasing nutrients back to soil where roots can absorb them again. This cycling retains nutrients within ecosystems. However, nutrients also leave forests through leaching, erosion, and harvest. Long-term forest productivity requires that nutrient inputs through weathering, nitrogen fixation, and atmospheric deposition balance these losses. Overharvesting can deplete soil nutrients, reducing future forest productivity. Phosphorus cycles differ from nitrogen cycles. Phosphorus does not have a significant atmospheric component. Phosphorus comes from rock weathering and cycles through soil, organisms, and back to soil. Phosphorus is often limiting in old, highly weathered tropical soils. Trees must invest heavily in mycorrhizal fungi to access phosphorus in these environments. Nitrogen cycles involve multiple transformations. Atmospheric nitrogen becomes available to plants through biological nitrogen fixation. Organic nitrogen in dead organisms converts to ammonia through decomposition. Ammonia can be taken up by plants or converted to nitrate through nitrification. Nitrate can be lost through leaching or denitrification that returns nitrogen to the atmosphere. Trees participate in these nitrogen transformations. Some trees host nitrogen-fixing symbionts. All trees take up nitrogen from soil. Nitrogen stored in wood remains sequestered until the wood decomposes, which can take decades or centuries. This long-term storage makes forests important nitrogen reservoirs in landscapes. Economic value of forests extends beyond timber. Forests provide watershed protection, preventing erosion and filtering water. They provide recreational opportunities, supporting tourism and contributing to human well-being. Non-timber forest products like mushrooms, medicinal plants, and nuts provide income for millions of people. Ecosystem services from forests, benefits that forests provide to humanity, are increasingly recognized and quantified. Carbon storage has economic value in carbon markets. Water provision and filtration have value to water users. Biodiversity conservation has value to society, even if it is difficult to price in monetary terms. Sustainable forest management attempts to balance multiple values and uses. Timber production can be managed to maintain forest cover and ecological function. Recreation can coexist with watershed protection. However, conflicts arise when different values or different stakeholders prioritize different forest uses. Indigenous peoples have managed forests sustainably for millennia. Traditional ecological knowledge includes understanding of forest regeneration, sustainable harvest levels, and relationships between forest health and human well-being. Incorporating indigenous knowledge into forest management can improve outcomes while respecting indigenous rights and cultures. Climate change presents unprecedented challenges to forests. Temperature increases, shift suitable habitat ranges poleward and upward in elevation. Precipitation pattern changes affect water availability. More frequent and intense droughts stress trees. Novel pest and disease pressures emerge as organisms expand into previously unsuitable areas. Tree species differ in vulnerability to climate change. Species with narrow climate tolerances face higher risk than generalist species. Species with limited dispersal ability may not keep pace with shifting climate zones. Species already at their cold or dry limits have nowhere to shift. Predicting which forests will persist and which will transform requires understanding these vulnerabilities. Some forests show signs of climate stress now. Increased tree mortality has been documented in Western North America, the Amazon, the Mediterranean, and elsewhere. Droughts that would have occurred once per century historically now occur more frequently, exceeding trees' drought tolerance. Rising temperatures increase water stress, even without precipitation changes. However, some forests may benefit from climate change, at least initially. Longer growing seasons can increase productivity in cold-limited boreal and alpine forests. Increased carbon dioxide can enhance photosynthesis. These potential benefits are uncertain and may be outweighed by stresses from drought, heat, and pest pressures. Forests will change under climate change regardless of human actions, but management can influence outcomes. Protecting forests from additional stressors like pollution and fragmentation improves their resilience. Reducing fire risk through fuel reduction maintains forest cover. Assisted migration, moving tree populations to match shifting climate conditions, remains controversial but may become necessary. Reforestation and afforestation, establishing forests on previously forested or non-forested lands, can help mitigate climate change through carbon sequestration. However, these efforts must consider multiple factors. Planting trees in grasslands or other non-forest ecosystems can harm those ecosystems and may not provide net climate benefits due to albedo changes. Appropriate species selection and site matching are critical for success. Urban forestry brings tree benefits to cities where most people live. Street trees cool neighborhoods, filter air pollution, reduce stormwater runoff, and provide aesthetic and psychological benefits. Green infrastructure incorporating trees is increasingly recognized as cost-effective compared to traditional grey infrastructure for managing urban heat and water. However, urban trees face challenges. Soil compaction, pollution, limited rooting space, and heat stress create hostile conditions. Salt damage from road de-icing kills many urban trees. Selection of appropriate species that can tolerate these stresses is critical. Investment in soil quality and adequate rooting space improves urban tree survival and growth. Water movement through trees represents one of the most remarkable processes in plant physiology. Water travels from soil through roots, up stems, and out through leaves, sometimes climbing more than 100 meters against gravity. This movement requires no pump, no moving parts, no energy input beyond solar heating that drives evaporation. The mechanism relies on cohesion and adhesion. Water molecules stick to each other through hydrogen bonding, creating continuous water columns inside xylem vessels. Water molecules also stick to xylem cell walls. As water evaporates from leaf surfaces, It pulls on water columns below, drawing water upward. This transpiration pull can lift water to great heights. The system works because water columns resist breaking under tension. However, extreme drought or freeze-thaw cycles can introduce air bubbles into xylem vessels, breaking water columns. This cavitation prevents water transport through affected vessels. Trees produce new xylem each year, partly to replace cavitated vessels. Leaf structure reflects the tension between maximizing photosynthesis and minimizing water loss. Stomata, small pores on leaf surfaces, open to allow carbon dioxide entry for photosynthesis. But open stomata also allow water vapor to escape. Trees constantly balance gas exchange and water conservation. Stomatal density varies among species and responds to environmental conditions. Desert plants typically have fewer stomata than rainforest plants. Trees grown under elevated carbon dioxide develop fewer stomata because each stoma can acquire more carbon when atmospheric carbon dioxide is higher. This adjustment occurs both within individual lifetimes and across evolutionary time. Leaf shape and size vary dramatically across species and climates. Large leaves maximize light capture but increase water loss and heat load. Small leaves lose less water and stay cooler, but require more investment in support tissue per unit area. Needle-shaped conifer leaves minimize water loss in cold or dry environments, but have relatively low photosynthetic capacity per leaf mass. Deciduous leaves fall annually in seasonal climates, while evergreen leaves persist for multiple years. Both strategies have advantages. Deciduous trees avoid winter maintenance costs and cold damage to leaves. Evergreen trees maintain photosynthetic capacity year-round and avoid the substantial cost of producing new leaves each spring. The transition from green leaves to autumn colors results from chlorophyll breakdown. Green chlorophyll normally masks other pigments in leaves. As chlorophyll breaks down in autumn, yellow carotenoid pigments and red anthocyanin pigments become visible. The intensity and timing of autumn color depends on species genetics, temperature, and light conditions. Why trees produce anthocyanins before leaf fall remains somewhat mysterious. These pigments require energy to produce. One hypothesis suggests anthocyanins protect leaf tissues during nutrient resorption, allowing trees to recover more nitrogen and phosphorus from leaves before dropping them. Another suggests anthocyanins protect against excess light that could damage photosynthetic machinery during breakdown. Nutrient resorption from senescing leaves represents a significant tree investment. Trees can recover 50 to 80% of nitrogen and phosphorus from leaves before leaf fall. This resorbed nitrogen and phosphorus is stored in woody tissue and recycled into new leaves the following spring. Efficient resorption provides competitive advantages in nutrient-poor environments. Bark serves multiple functions beyond structural support. It protects inner tissues from mechanical damage, insect attack, pathogen infection, and temperature extremes. It prevents water loss from the trunk. It sometimes contains photosynthetic tissue in young stems. Bark thickness and characteristics vary dramatically among species. Some trees have very thin bark that provides minimal fire protection. Others, like ponderosa pine and giant sequoia, develop bark more than a foot thick that insulates cambium from ground fires. Cork oak bark can be harvested repeatedly without killing the tree, providing sustainable cork production for centuries from individual trees. Bark patterns are often species-specific, allowing tree identification, even in winter when leaves are absent. Smooth bark, furrowed bark, shaggy bark, plated bark, and exfoliating bark all reflect different growth patterns and adaptation strategies. Some species change bark characteristics dramatically with age, starting with smooth bark and developing deep furrows as trunks expand. Lenticels, small pores in bark, allow gas exchange. Living tissues inside bark require oxygen and produce carbon dioxide. lenticels provide pathways for these gases to diffuse between inner tissues and atmosphere. In very wet or flooded conditions, lenticel function becomes critical for tree survival. Branches and stems respond to mechanical stress through reaction wood formation. When trees lean or branches are loaded by weight, trees produce wood with different properties than normal wood. This reaction wood generates forces that pull or push stems and branches, helping maintain structural integrity and orientation. In conifers, compression wood forms on the lower side of leaning stems, pushing the stem upward. In angiosperms, tension wood forms on the upper side of leaning stems, pulling the stem upward. These reaction woods have different cellular structure and chemical composition than normal wood, making them identifiable in cross-sections. Wood density varies within individual trees and among species. Wood formed early in the growing season. Spring wood is less dense than wood formed later, summer wood. This density difference creates visible growth rings. Density also varies from pith to bark in many species, with wood becoming denser with age. Wood density influences multiple tree characteristics. Dense wood is stronger and more rot-resistant, but more costly to produce. Light wood allows faster height growth with less investment, but provides less structural support per volume. Different species balance these trade-offs differently, depending on their ecology. Heartwood and sapwood differ in function and composition. Sapwood, the outer wood layer, actively transports water and stores nutrients. Heartwood, the inner wood, no longer transports water but provides structural support. During heartwood formation, cells die and trees deposit chemical compounds that resist decay and darken the wood. Heartwood formation is why many old trees are hollow yet still living. The hollow interior was heartwood that eventually decayed. As long as sapwood remains intact, water transport continues and the tree survives. The hollow actually provides some advantages, reducing weight while maintaining structural strength, though it also creates vulnerability to breakage. Wood decomposition returns carbon and nutrients to soil, but also provides habitat. Dead wood, whether standing or fallen, supports remarkable biodiversity. Fungi initiate decomposition, breaking down complex wood polymers. Insects colonize decaying wood, exploiting this resource and providing food for birds and mammals. Salamanders and small mammals use cavities in decaying logs. Decomposition rates vary by wood species, size, and environmental conditions. Dense wood decomposes slowly. Large logs can persist for decades or centuries. In dry environments, decomposition is very slow. In moist tropical environments, decomposition can be remarkably rapid, with fallen trees disappearing within a few years. Coarse woody debris, the technical term for dead wood in forests, was historically removed from managed forests as waste. However, recognition of its ecological importance has changed management. Retaining dead wood supports biodiversity and contributes to nutrient cycling. Balance is needed to maintain forest health while providing wildlife habitat. Trees as organisms are incredibly complex, but tree communities create even more complexity. Forest structure includes multiple layers. Canopy trees form the uppermost layer, receiving full sunlight. Subcanopy trees form an intermediate layer. Understory shrubs and herbs occupy the forest floor. These layers create diverse light, temperature, and moisture conditions within a single forest. Gap dynamics drive forest regeneration in many systems. When canopy trees fall, they create gaps where light reaches the forest floor. These gaps allow shade-intolerant species to establish and grow. Over time, the gap fills and the forest returns to closed canopy. This process creates a mosaic of different aged patches across the landscape. Old-growth forests develop characteristics that young forests lack. Large living trees and large dead trees create structural complexity. Multiple canopy layers provide diverse habitat. Accumulated coarse woody debris supports decomposer communities. Old trees with cavities provide nesting sites for cavity-using birds and mammals. These characteristics develop over centuries and are difficult or impossible to restore quickly once lost. The concept of climax forest, a stable endpoint of succession, has been largely abandoned. Forests continue changing even after reaching old-growth condition. Disturbances like storms, droughts, and pest outbreaks constantly create heterogeneity. Individual trees die and are replaced. Species composition shifts as climate changes. Forests are best understood as dynamic systems rather than static climaxes. Succession, the process of vegetation change following disturbance, follows general patterns but varies in details. Primary succession occurs on bare substrate like volcanic rock or sand dunes where no soil exists initially. Pioneer species colonize these harsh sites, beginning soil formation. Secondary succession occurs after disturbance in areas with existing soil, proceeding more rapidly than primary succession. Early successional species typically are fast-growing, light-demanding, and short-lived. They produce abundant seeds that disperse widely. They establish quickly after disturbance, but cannot persist in their own shade. Late successional species grow more slowly, tolerate shade, and live longer. They eventually replace early successional species, unless disturbance resets succession. However, arrested succession can occur when conditions prevent late successional species establishment. Repeated fires can maintain grasslands that would otherwise succeed to forest. Browsing by deer or other herbivores can prevent tree seedling establishment, maintaining shrublands. Human activities like agriculture obviously arrest or reverse succession. Forest edges differ from forest interiors in microclimate and species composition. Edge effects extend tens to hundreds of meters into forests, depending on aspect, prevailing winds, and surrounding land use. Edges experience more wind, more light, greater temperature variation, and lower humidity than interiors. These changed conditions favor different species than forest interiors support. Forest fragmentation increases edge effects and reduces interior forest habitat. Small forest patches have proportionally more edge than large patches. This matters for species requiring interior forest conditions. Birds that nest in interior forests often abandon small fragments due to edge effects and increased predation. Maintaining large forest blocks becomes important for biodiversity conservation. Corridors connecting forest fragments can facilitate animal movement and gene flow between isolated populations. However, corridor effectiveness depends on width, composition, and landscape context. Narrow corridors may function as edges rather than interior habitat. Poorly designed corridors can potentially spread fire or disease. Corridor conservation requires careful planning. Trees modify their environment, creating positive feedbacks that maintain forest cover. Forests shade ground, reducing temperature and maintaining humidity. They moderate wind. They add organic matter to soil, improving water retention and nutrient availability. These conditions favor tree seedling establishment, helping maintain forest over time. However, these feedbacks can be broken. Severe disturbance or climate change can push forests past tipping points where conditions no longer favor tree regeneration. Grassland or shrubland replaces forest. Restoring forest after crossing such tipping points becomes much more difficult than maintaining existing forest. Alternative stable states occur where multiple vegetation types can persist under the same climate, depending on history and feedbacks. Fire-maintained grasslands and forests can both persist in some climates, depending on fire frequency. Shrublands and forests can both be stable in Mediterranean climates. Understanding these alternative states matters for forest conservation and restoration. Soil properties strongly influence forest composition and productivity. Soil depth determines rooting volume and water storage capacity. Shallow soils support shorter trees and less biomass than deep soils. Soil texture affects water and nutrient holding capacity. Sandy soils drain quickly but hold few nutrients. Clay soils hold water and nutrients, but can restrict root penetration. Soil pH affects nutrient availability. Most nutrients are most available at near neutral pH. Very acidic or alkaline soils limit availability of certain nutrients, constraining which species can thrive. Some tree species acidify soils, creating positive feedbacks that maintain their dominance. Others tolerate or require alkaline conditions and dominate limestone-derived soils. Parent material, the rock from which soil forms, influences forest types. Granite-derived soils are typically acidic and nutrient-poor, supporting species adapted to these conditions. Basalt-derived soils are often more fertile, supporting higher productivity forests. Limestone creates alkaline conditions with high calcium availability. Soil organisms are as diverse and important as above-ground forest organisms. Bacteria and fungi decompose organic matter and cycle nutrients. Protozoa and nematodes consume bacteria, releasing nutrients in plant-available forms. Arthropods fragment, litter, and mix soil layers. Earthworms can dramatically alter soil structure and nutrient dynamics where present. The ratio of bacteria to fungi in soil varies among forest types and influences ecosystem function. Fungal-dominated soils occur in coniferous forests and are associated with slower decomposition and nutrient release. Bacterial-dominated soils occur more in deciduous forests and support faster nutrient cycling. These differences affect forest productivity and response to disturbance. Mycorrhizal fungi deserve particular attention given their ubiquity and importance. More than 90% of plant species, including almost all trees, form mycorrhizal associations. The fungi receive carbon from trees and provide nutrients and water in return. This exchange is fundamental to forest ecosystem function. Two main mycorrhizal types exist. Ectomycorrhizal fungi form sheaths around root tips and hyphae that penetrate between, but not into root cells. They associate mainly with conifers and temperate hardwoods. Arbuscular mycorrhizal fungi penetrate root cells, forming tree-like structures called arbuscules. They associate with most tropical trees and some temperate species. These different mycorrhizal types have different ecological impacts. Ectomycorrhizal networks can be more extensive, potentially connecting more trees and transporting resources over longer distances. Arbuscular mycorrhizal fungi produce glomalin, a sticky protein that helps stabilize soil aggregates and store carbon. Both types are critical for forest health. Mycorrhizal fungi vary in their efficiency at acquiring different nutrients. Some species excel at mobilizing organic nitrogen. Others access mineral forms preferentially. Some tolerate or require acid soils. Others thrive in neutral conditions. This diversity allows trees to associate with different fungi depending on soil conditions, maximizing nutrient acquisition across varied environments. Trees can associate with dozens of different mycorrhizal fungal species simultaneously. This diversity provides insurance. If some fungi are affected by environmental changes, others may compensate. It also allows exploitation of different soil microsites. Fine roots with different fungal partners can access different nutrient sources within centimeters of each other. Fungal community composition changes with forest age and development. Pioneer trees often associate with generalist fungi that produce abundant spores and colonize disturbed sites readily. Late successional trees associate with specialist fungi that produce fewer spores but form more extensive networks. These shifts in fungal communities parallel shifts in tree species composition during succession. Climate affects mycorrhizal fungi as directly as it affects trees. Drought stresses fungi as well as trees, potentially disrupting the symbiosis when it is most needed. Warming temperatures may favor some fungal species over others. Nitrogen deposition can reduce mycorrhizal colonization because trees invest less in fungal partnerships when nitrogen is abundant. Understanding mycorrhizal ecology has practical applications. Inoculating tree seedlings with appropriate fungi improves establishment success, particularly in harsh environments or disturbed sites. Maintaining diverse fungal communities in managed forests supports tree health and resilience. Avoiding practices that damage soil fungal communities, like excessive tillage or removal of organic matter, preserves this critical resource. Pollination in trees differs from pollination in herbaceous plants in some respects, but follows similar principles. Trees must transfer pollen from male reproductive structures to female structures. Wind pollination and animal pollination are the two main strategies, each with distinct characteristics and trade-offs. Wind pollination dominates among conifers and some temperate deciduous trees. Wind-pollinated trees produce enormous pollen quantities to compensate for the randomness of wind dispersal. Pollen is small, lightweight, and smooth, allowing efficient wind transport. Male and female flowers lack showy petals or nectar, since there is no need to attract animals. Animal pollination occurs in many flowering trees, particularly tropical species. Insects, birds, and bats serve as pollinators. Animal-pollinated flowers produce nectar to reward visitors, and often have colorful petals or strong scents to attract them. Pollen is larger and stickier than wind-pollinated pollen, adhering to animal visitors. Some trees show remarkable pollinator specialization. Certain fig species are pollinated by specific fig wasp species, each fig species having its own wasp pollinator. The wasps cannot complete their life cycle without the fig, and the fig cannot reproduce without the wasp. Such specialized mutualisms are vulnerable to disruption if either partner declines. Flowering time matters for pollination success. Trees must flower when pollinators are active or when weather conditions favor wind pollination. Flowering too early risks frost damage to flowers. Flowering too late may miss optimal pollinator activity. Climate change is shifting flowering times, potentially creating mismatches with pollinators. Seed production requires substantial investment. Trees allocate significant resources to flowers, fruits, and seeds. This investment competes with growth and defense. Trees must balance current reproduction against future survival and reproduction. This life history trade-off influences how resources are allocated across the lifespan. Predispersal seed predation, consumption of seeds before they disperse from parent trees, can dramatically reduce reproductive success. Insects that lay eggs in flowers or developing fruits can destroy most or all of a tree's seed crop. Squirrels and birds harvest seeds from cones and fruits. These losses select for defensive strategies like toxic seed coats or masting. Mast seeding, synchronized heavy seed production across multiple years or regions, overwhelms seed predators. In mast years, so many seeds are produced that predators cannot consume them all. In non-mast years, low seed production reduces predator populations. This strategy works at population level, even though individual trees sacrifice reproduction in low years. Post-dispersal seed predation continues after seeds reach the ground. Rodents, birds, and insects harvest seeds from forest floors. This predation creates density-dependent mortality. Areas near parent trees typically have high seed density, but also attract more seed predators, reducing seedling establishment immediately under parent trees. This creates the Jansen-Connell effect, where seedling survival is lowest near parent trees and increases with distance. The effect results from specialized seed predators and pathogens that concentrate near parent trees. This distance-dependent mortality promotes tree diversity by preventing any species from locally dominating. Seed dormancy allows seeds to wait for favorable conditions. Some seeds germinate immediately if conditions are suitable. Others require exposure to cold, scarification, or other treatments before germinating. This dormancy spreads germination over time, bet hedging against environmental uncertainty. Seed banks, accumulations of viable seeds in soil, create populations ready to colonize disturbed sites. Some species maintain persistent seed banks, with seeds remaining viable for years or decades. Others have transient seed banks, where seeds germinate within a year or die. Seed bank characteristics influence forest regeneration after disturbance. Genetic diversity within tree species matters for population persistence. Genetic variation allows adaptation to environmental changes. Populations with low genetic diversity are more vulnerable to diseases, pests, and environmental shifts. Maintaining connectivity between populations preserves gene flow that sustains diversity. Trees have long generation times compared to annual plants or animals. Adaptations that take only years in short-lived organisms may take centuries in long-lived trees. This slow evolutionary rate means trees may not adapt quickly enough to rapid climate change, raising conservation concerns. However, trees also show phenotypic plasticity, the ability to adjust to environments without genetic change. Individual trees can modify growth patterns, leaf characteristics, and other traits in response to conditions. This plasticity provides some capacity to cope with changing environments, even without genetic adaptation. Hybridization between closely related tree species occurs more commonly than once thought. Hybrid zones form where species ranges overlap. Hybrids sometimes show traits intermediate between parents. In other cases, hybrids exhibit novel trait combinations or exceed parents in vigor. Hybridization can introduce genetic variation and facilitate adaptation. However, hybridization also raises conservation concerns. Rare species may hybridize with common relatives, losing distinct characteristics. Introduced species may hybridize with natives, genetically swamping local populations. Managing these situations requires balancing preservation of distinct lineages against the role of hybridization in generating diversity. Tree physiology responds to light quality and quantity. Photosynthetically active radiation, wavelengths between 400 and 700 nanometers, drives photosynthesis. Red and blue wavelengths are absorbed most effectively. Green wavelengths are reflected, which is why leaves appear green. Light outside this range does not contribute to photosynthesis. Photosynthetic efficiency varies with light intensity. At low light, photosynthesis increases linearly with increasing light. At moderate light, photosynthesis saturates, with additional light providing no additional benefit. At very high light, photosynthesis may decline due to photoinhibition, where excess light damages photosynthetic machinery. Shade tolerance reflects species' ability to photosynthesize and survive at low light. Shade-tolerant species have lower light compensation points, the light level where photosynthesis equals respiration. They can survive in forest understories where shade-intolerant species cannot. However, shade-tolerant species typically grow more slowly than shade-intolerant species at high light. This creates a growth-survival trade-off. Shade-intolerant species grow rapidly when light is available but cannot persist in shade. Shade-tolerant species grow slowly but survive shade that kills shade-intolerant species. These different strategies allow coexistence through temporal and spatial niche partitioning. Carbon allocation within trees follows patterns that change with tree size and age. Seedlings allocate carbon primarily to leaves to maximize photosynthetic capacity and to roots for water and nutrient acquisition. As trees grow, an increasing proportion goes to stem and branch wood for structural support. Mature trees allocate substantial carbon to reproduction. The carbon allocation puzzle asks how trees decide where to allocate limited carbon. Multiple factors influence allocation, including resource availability, growth demands, defense needs, and reproductive opportunities. Trees must balance these competing demands, and optimal allocation patterns change with conditions and life stage. Storage of carbohydrates provides insurance against future carbon deficits. Trees store starch and sugars in roots, stems, and branches. These reserves support leafing out in spring before new leaves can photosynthesize. They support recovery from damage or stress. They can sustain trees through temporary periods when respiration exceeds photosynthesis. Carbon storage capacity varies among species. Some trees store little, depending on continuous photosynthesis, to meet demands. Others store substantial reserves that allow persistence through years of poor growing conditions. These storage strategies reflect species ecology and the environments they evolved in. Respiration consumes substantial proportions of photosynthetically fixed carbon. Growing tissue requires energy for biosynthesis. Maintenance respiration sustains existing tissue. Root respiration provides energy for nutrient uptake and growth. Total respiration can consume 40 to 70% of photosynthesis in trees, leaving the remainder for growth and storage. Respiration rates increase with temperature, creating concerns under climate warming. If respiration increases faster than photosynthesis with warming, trees may experience carbon deficits. This could reduce growth or increase mortality. Understanding temperature responses of photosynthesis and respiration is critical for predicting forest responses to warming. Hydraulic architecture, the structure of water transport systems, constrains tree form and function. Water must reach all living tissues. The path from roots to leaves becomes longer as trees grow taller. Resistance to flow increases with path length. Eventually, water transport limitations constrain further height growth. Xylem vessel diameter influences water transport efficiency. Wide vessels transport more water per unit pressure difference, but are more vulnerable to cavitation. Narrow vessels are safer, but require more vessels to transport the same water volume. Trees balance these trade-offs differently, producing wood with different vessel characteristics. Taper. The decrease in stem diameter from base to crown reflects hydraulic needs. Maintaining sufficient conducting area throughout the tree's height requires tapering stems. Optimal taper balances hydraulic conductance against mechanical stability. Trees that develop more taper typically have less cavitation in upper branches. Branch architecture reflects hydraulic constraints and mechanical requirements. Branches must conduct water to leaves while supporting their own weight. Wide branch angles increase mechanical stability, but create sharper turns that increase hydraulic resistance. Narrow angles reduce hydraulic resistance, but decrease mechanical stability. Trees modify their architecture in response to mechanical stress. Wind sway promotes stem thickening and branch reduction. Heavy fruit or snow loads trigger similar responses. This mechanical acclimation allows trees to optimize structures for their specific mechanical environments, though the optimization occurs slowly over years. Roots explore soil for resources and anchor trees against toppling. Root system architecture varies among species and with soil conditions. Tap roots penetrate deeply. Lateral roots spread horizontally. Fine roots with high surface area to volume ratios absorb water and nutrients. Coarse roots provide structural support and transport. Root-shoot ratio. The proportion of biomass in roots versus shoots varies systematically with environment. Dry environments favor high root-shoot ratios. Fertile environments allow low root-shoot ratios. This makes functional sense. When water is limiting, invest in roots to access more water. When nutrients are abundant, invest in shoots to capture light. Root grafting between trees creates structural and physiological connections. Natural grafts form where roots grow in contact and tissues fuse. These connections allow water, nutrient, and chemical signal transfer between trees. In some cases, stumps without leaves persist for decades, receiving sugars through root grafts with neighboring living trees. Root exudates, organic compounds released from roots into soil, serve multiple functions. They can attract beneficial microbes, suppress competitors, mobilize nutrients, or defend against pathogens. The diversity and quantity of root exudates vary among species, influencing soil communities and plant interactions. Allelopathy, chemical suppression of competitors, occurs in some tree species. Black walnut produces juglone, toxic to many other plants. Eucalyptus produces compounds that inhibit germination and growth of potential competitors. These chemicals provide competitive advantages by reducing competition for light, water, and nutrients. However, demonstrating allelopathy in natural systems is challenging. Many compounds that are allelopathic in laboratory tests occur at lower concentrations in field soils. Microbes may decompose potential allelopathic compounds. Effects attributed to allelopathy may actually result from resource competition. Careful experiments are required to distinguish these possibilities. Nitrogen fixation by symbiotic bacteria provides enormous advantages in nitrogen-poor soils. Trees in genera, like Alnus, Robinia, and Casuarina, host actinomycete bacteria in root nodules. These bacteria convert atmospheric nitrogen to ammonia that trees can use. This nitrogen enriches soil as leaves and roots decompose, benefiting neighboring plants. Nitrogen-fixing trees often dominate early succession in nitrogen-poor sites. They grow rapidly despite low soil nitrogen, eventually improving soil nitrogen through litter deposition. This facilitation allows later succession by species that require higher soil nitrogen but cannot fix their own. The nitrogen fixers may eventually be replaced by non-fixers that compete effectively once nitrogen is abundant. However, nitrogen fixation is energetically expensive. Trees must provide carbohydrates to bacteria, reducing carbon available for growth. This cost means nitrogen-fixing trees are poor competitors in nitrogen-rich environments, where non-fixers can access soil nitrogen without supporting symbionts. Nitrogen fixers dominate mainly where their unique capability provides advantage. Phosphorus acquisition strategies vary among tree species. Some produce organic acids that dissolve phosphorus from mineral forms. Others invest heavily in mycorrhizal fungi that search large soil volumes. Some produce specialized root structures called cluster roots, with extremely high root surface area and exudation rates. Trees in ancient, highly weathered tropical soils face particular phosphorus challenges. Most phosphorus is locked in unavailable forms. Trees must invest enormous resources in phosphorus acquisition, slowing growth. Species that evolved in these environments have efficient phosphorus use, producing more tissue per unit phosphorus than species from fertile temperate regions. The concept of nutrient use efficiency describes how much growth a tree achieves per unit nutrient. Trees with high nutrient use efficiency produce more biomass per unit nitrogen or phosphorus than trees with low efficiency. This efficiency provides advantage in nutrient-poor soils, but may limit maximum growth rate even when nutrients are abundant. Trees also vary in their resource acquisition strategies more broadly. Some have acquisitive strategies, growing rapidly, with short-lived tissues and high resource turnover. Others have conservative strategies, growing slowly with long-lived tissues and low resource turnover. These strategies represent alternative ways to succeed in different environments. The leaf economic spectrum describes global patterns in leaf traits. Acquisitive species have high photosynthetic rates, high nitrogen concentrations, thin leaves, and short leaf lifespans. Conservative species have low photosynthetic rates, low nitrogen, thick leaves, and long leaf lifespans. These traits co-vary globally across thousands of species. This spectrum reflects fundamental trade-offs. Producing high-quality leaves that photosynthesize rapidly costs substantial nitrogen and phosphorus. If these nutrients are abundant and leaves face low risk of damage, the acquisitive strategy succeeds. If nutrients are scarce or leaves face high damage risk, the conservative strategy of producing protected, long-lived leaves succeeds despite lower photosynthetic rates. Wood economic spectrum describes analogous trade-offs in wood traits. Acquisitive species produce low-density wood that allows rapid growth but provides less structural support and durability. Conservative species produce high-density wood that grows slowly but provides better support and durability. These strategies also trade off growth rate against tissue longevity. Understanding these spectrums helps predict tree responses to environmental changes. Acquisitive species should increase growth more under elevated carbon dioxide or fertilization. Conservative species should better tolerate drought or nutrient limitation. However, predictions must account for interactions among multiple environmental factors. Invasive tree species illustrate ecological principles while creating conservation challenges. Successful invaders often are acquisitive species that grow rapidly and reproduce prolifically. They typically face fewer enemies in introduced ranges than in native ranges, releasing them from regulation. This enemy release allows invasion success. However, not all introduced trees become invasive. Most fail to establish persistent populations. Understanding what determines invasion success matters for predicting and preventing future invasions. Traits associated with invasion success include rapid growth, prolific seeding, broad environmental tolerance, and long-distance dispersal. Invasive trees can dramatically alter invaded ecosystems. They may outcompete native trees, reducing diversity. They may change fire regimes, nutrient cycles, or water availability. Managing established invasions is challenging and expensive. Prevention through screening potential introductions is more effective than attempting control after establishment. Trees interact with other organisms at every life stage. Seeds are eaten by predators and dispersed by mutualists. Seedlings are browsed by herbivores and facilitated by shade-casting neighbors. Mature trees are attacked by insects and pathogens, while supporting epiphytes and providing cavities for animals. Even dead trees provide habitat and resources for decomposers and cavity users. These interactions span trophic levels. Trees are primary producers, converting solar energy to chemical energy. Herbivores consume tree tissues, transferring energy to higher trophic levels. Predators and parasites regulate herbivore populations. Decomposers return nutrients to soil. These interactions create food webs of remarkable complexity. Keystone species, organisms whose impacts exceed what would be expected from abundance, can strongly influence forest communities. Beavers, by damming streams and flooding areas, kill trees and create wetlands. This transforms landscape structure, creating habitat for species that could not otherwise occur. Removing beavers eliminates wetlands and the species they support. Ecosystem engineers, organisms that modify physical environments, include many tree species. Trees that produce deep shade suppress understory vegetation. Those that produce easily decomposed litter create nutrient-rich surface soils. Those that produce recalcitrant litter create nutrient-poor acidic soils. These engineering effects persist long after individual trees die. Foundation species, organisms that strongly affect ecosystem function, include many dominant forest trees. Loss of foundation species can cause ecosystem regime shifts. Chestnut blight eliminated American chestnuts as dominants across eastern North America. The forests that replaced chestnut-dominated forests have different structure, composition, and function. Forests provide cultural and spiritual values that are difficult to quantify, but deeply important. Sacred groves in many cultures preserve forest patches for religious purposes. These preserved areas sometimes contain biodiversity found nowhere else. Individual ancient trees become cultural landmarks, connecting communities to history. Traditional ecological knowledge about forests, developed over generations, provides valuable insights. Indigenous peoples have managed forests sustainably for millennia. Understanding seasonal patterns, indicator species, and appropriate harvest levels. This knowledge complements scientific understanding and can improve forest conservation and management. Forest bathing, the practice of spending time in forests for health benefits, is supported by growing evidence. Time in forests reduces stress hormones, lowers blood pressure, and improves mood. The mechanisms may involve phytoncides, airborne compounds released by trees, though simply being in natural settings provide psychological benefits. Urban residents often have limited forest access, creating environmental justice issues. Lower-income neighborhoods typically have less tree cover than wealthier neighborhoods. This creates disparities in tree benefits like cooling, air quality improvement, and stress reduction. Addressing these inequities requires intentional efforts to increase tree cover in underserved areas. Citizen science engages public volunteers in forest monitoring and research. Programs like phenology observations, where volunteers report timing of leafing and flowering, generate valuable data on climate change responses. Tree identification apps encourage learning about local trees. Volunteer forest inventories contribute to understanding urban forest composition. Education about forests builds appreciation and support for conservation. When people understand forest ecology and values, they are more likely to support forest protection. School programs that bring children into forests create early connections with nature. These experiences can shape lifelong attitudes and behaviors. Forest restoration can repair damaged ecosystems and provide multiple benefits. Reforesting degraded lands sequesters carbon, prevents erosion, and creates wildlife habitat. Restoration is not simply planting trees, however. Appropriate species selection, site preparation, and ongoing maintenance influence success. Restored forests may take decades to centuries to develop old-growth characteristics. Passive restoration, allowing natural regeneration without intervention, works in some circumstances. When seed sources are nearby and soil conditions are suitable, forests can regenerate naturally. This approach costs less than active planting and can create forests with greater diversity and local adaptation. However, passive restoration requires patience and may not work if conditions prevent natural regeneration. Assisted migration, moving tree populations toward cooler or wetter climates to match projected future conditions, remains controversial. Proponents argue it may be necessary for trees to keep pace with rapid climate change. Opponents worry about introducing trees where they could become invasive or disrupt existing ecosystems. The debate continues as climate velocity exceeds many species' natural migration rates. Provenance, the source location of seeds or seedlings, matters for restoration and forestry success. Trees evolved adaptations to local conditions. Moving trees long distances can create maladaptation where introduced populations do not thrive. However, small movements may introduce genetic variation that improves resilience. Finding optimal provenance remains an active research area. Seed zones map geographic areas within which seeds can be moved without risking maladaptation. These zones consider climate, elevation, and other factors influencing local adaptation. Staying within seed zones generally produces best results. However, climate change raises questions about whether historical seed zones remain appropriate or whether adjustments are needed. Traditional forestry focused primarily on timber production. Practices like clear-cutting, even-aged management, and planting monocultures maximized wood volume, but reduced structural diversity and habitat quality. Recognition of forests' multiple values has led to more diversified management approaches, balancing timber production with conservation objectives. Sustainable yield forestry aims to harvest timber continuously while maintaining forest cover and function. Rotation length, the time between harvest and reharvest, influences forest age structure and composition. Longer rotations allow development of more old-growth characteristics. Shorter rotations maximize volume production, but create young forests lacking complexity. Selection harvesting removes individual trees while maintaining forest canopy, creating smaller-scale disturbance than clear-cutting. This approach better maintains forest interior conditions and structural diversity. However, it may not suit species requiring high light for regeneration. Different harvest systems suit different forest types and objectives. Certification programs like FSC and SFI set standards for responsible forest management. Certified forests must meet criteria for conservation, community relations, and sustainable harvests. While certification is not perfect, it provides incentives for improved practices and helps consumers identify products from responsibly managed forests. Old-growth forest protection has been contentious because old growth often contains largest trees with most timber value. However, old-growth characteristics develop only over centuries and cannot be quickly replaced. Protection of remaining old growth prevents loss of irreplaceable forests, while allowing management of younger forests for timber and restoration. Climate-smart forestry attempts to manage forests in ways that increase resilience to climate change, while maintaining carbon storage and other values. Practices might include increasing diversity, protecting genetic variation, reducing other stressors, and adapting management as conditions change. This adaptive approach acknowledges uncertainty while taking action. The future of forests depends on countless interacting factors. Climate change will shift where different forest types can exist. Human land use will determine how much land remains available for forests. Our harvest intensity will influence forest age and structure. Pest and pathogen pressures will continue evolving. All these factors interact in complex, sometimes unpredictable ways. Yet trees themselves are not passive recipients of change. They adapt, migrate, and modify their environments. They interact with evolving communities of fungi, insects, birds, mammals, and other organisms. These dynamic interactions have persisted through previous periods of rapid environmental change. While the current pace of change is unprecedented in human timescales, trees have survived challenges throughout their evolutionary history. The relationship between humans and trees continues evolving. We depend on forests for materials, ecosystem services, and psychological well-being. Trees persist despite, and sometimes because of, human activities. Understanding this relationship matters for both human flourishing and forest conservation. The two are not opposed but interdependent. As this exploration winds toward conclusion, what persists is less the specific facts themselves than a sense of trees' complexity. These are not simple organisms, but sophisticated systems, integrating responses to environment, coordinating growth and development, interacting with countless other species, and influencing ecosystems from local to global scales. Trees also provide lessons about patience and time. A seedling germinating today might live centuries, growing slowly, responding to environmental changes, producing seeds that establish the next generation. This long-term perspective contrasts sharply with human planning horizons measured in years or decades. Climate change presents unprecedented challenges for trees and forests. Warming temperatures, shifting precipitation patterns, more frequent extreme weather, and novel pest and disease pressures all threaten tree survival. Species adapted to current conditions may find those conditions no longer exist within their natural range. Migration rates may not keep pace with climate velocity, yet trees and forests have persisted through dramatic environmental changes throughout Earth history. They survived ice ages, warm periods, meteorite impacts, and countless regional catastrophes. While individual species may face extinction and forest distributions will certainly shift, trees as a group will continue finding environments where they can survive and grow. The future of forests depends partly on human decisions. Protecting existing forests prevents carbon emissions and maintains biodiversity. Restoring degraded lands with trees sequesters carbon and repairs ecosystem function. Managing working forests sustainably provides wood products while maintaining forest ecological functions. Urban forestry brings tree benefits to cities where most people live. These actions require recognizing forests' value beyond timber. Forests provide clean water, prevent erosion, support wildlife, produce oxygen, sequester carbon, moderate climate, and contribute to human well-being. Economic analyses increasingly attempt to quantify these ecosystem services, putting monetary values on benefits forests provide. But some forest values resist quantification. The aesthetic experience of walking through forests, the peace found in contemplating ancient trees, the wonder of understanding how forest ecosystems function. These values are real, even if difficult to measure or price. Trees remind us that growth need not be rapid to be valuable, that success can emerge from patience and persistence, that complexity can arise from interactions among many simple parts. They demonstrate strategies for surviving in changing environments. Flexibility in response, diverse bet hedging approaches, cooperation alongside competition, investment in future generations. As sleep approaches and consciousness dims, let these thoughts about trees settle gently. No urgency, no need to remember details, simply an appreciation that trees exist, that they shape the world in profound ways, that they demonstrate alternative ways of living that complement rather than compete with human approaches. The forests will continue regardless of human understanding or appreciation. Trees will keep growing, communicating through mycorrhizal networks, responding to their environments, producing seeds, and slowly transforming carbon dioxide into living tissue. These processes operated long before humans evolved and will continue long after humanity passes. There is comfort in that continuity, that stability, that persistence. The universe contains patterns that endure across timescales, exceeding human comprehension. Trees participate in those patterns, linking atmospheric chemistry to soil biology, connecting organisms across multiple scales, bridging microscopic and macroscopic phenomena. Better sleep, a lighter day.